The story of human gene editing. Scientists are editing genes inside a living person for the first time, in an attempt to cure genetic disorder. The gene editing technology is controversial, but holds the potential for curing serious conditions by removing damaging genes and replacing them with properly functioning ones. Earlier this year, American scientists used CRISPR to delete disease-causing genetic mutations from human embryos. They allowed the embryos to develop for a few days, to watch their progress, before terminating them. The researchers at UCSF Benioff Children's Hospital Oakland dripped new genes into the patient that will replace mutated ones causing his illness, known as Hunter syndrome. Hunter syndrome is a progressive disorder that is linked to the absence of a certain enzyme, because a genetic mutation prevents the body from producing it. Without that enzyme, hypertrophy of the face, vocal cords and in the patient's airway occurs, which affects the breathing. The disorder can lead to brain edema, thickened skin, skin growths and other issues that lead to complications like hearing and vision loss, recurring infections and heart problems. Hunter syndrome patients can be treated with the missing enzyme on a weekly basis, but it won't stop damage to their brains, or reverse damage that has already been caused, and is a costly avenue. Brian Maddox, the first person to try out this new treatment, is willing to take the risk. The 44-year-old man has been waiting for this treatment that can potentially cure him. The experts will test the therapy on him first, but, if successful, eventually the treatment may be used on significantly younger patients who have not experienced as much tissue damage. CRISPR has become almost synonymous with the phrase gene editing tool in public consciousness. But this attempt is using a different tool that relies on proteins called zinc finger nucleases that bind to DNA and allow scientists to target a specific location in the genome. Here the original, harmful gene is removed and replaced with a new, disease-free one. The new gene will make the enzyme that is currently lacking. There are different forms of gene therapy, but the kinds that involve editing a person's DNA have been controversial because of the potential for something to go wrong and people's worries about how they will be used in the future. Scientists have been concerned about whether changing one element of DNA could trigger other unforeseen changes that are potentially detrimental. And at least one study has suggested that editing genes with CRISPR could cause many unpredictable genetic mutations, based on an analysis of small changes found in the genomes of mice that had been treated with gene therapy to cure blindness. The idea for critics is that even though we are constantly learning more about human genetics, we do not have a full enough understanding to predict all the ways DNA could behave once we start tweaking genes. Another criticism of advanced gene editing technology is that it could be used on embryos and fetuses to make designer babies, with parents choosing their children's hair and eye color. The main advantage of gene editing is devastating genetic diseases and birth defects can be erased while a baby is still developing. Thank you.